Okay, so in this part, I'm going to discuss what is hematopoiesis. And of course, hematopoiesis is simply the formation of blood cellular components in your body. And more simply, it is the process that manufactures blood cells. So hematopoiesis is going to occur in the embryo, all the way through adulthood. And so in the embryo, it starts in the yolk sac, and then it moves on to the liver. And then after birth and in adulthood, it is the bone marrow that is the sole source of making your erythrocytes and granulocytes. Now, overall, with respect to hematopoiesis, it begins with what we now know as a pluripotent stem cell. And this pluripotent stem cell is what we refer to as self-renewing. So in essence, the stem cells can make themselves, they can make more stem cells. Um, and then so when the stem cells get some sort of signal, and the signal is going to be outside telling the stem cells what kind of cells they need. It might be a red blood cell, it might be a platelet, it might be a B cell, it might be a T cell. There's going to be some sort of growth factors or stimulating factors or cytokines. And then they're going to get the message that now we need to make a different cell type. And this cell type that this pluripotent stem cell within the bone marrow will make is going to be a myeloid lymphoid progenitor cell. Now this cell is a step down from a pluripotent stem cell, but it is a stem cell in the sense that it can form multiple subtypes of cells. And so this myeloid lymphoid progenitor cell will produce two other progenitor cells, and they are going to be the common lymphoid progenitor cell and it's going to be the common myeloid progenitor cell. Now these cells are also two steps down from your pluripotent stem cell, but they too can become multiple cells. And so there's going to be another series of growth factors, stimulating factors. This is just growth factors, stimulating factors, plus cytokines. And then these cells, oops, I'll make this a different color. Growth factors, stimulating factors, cytokines. And then these are going to become our myeloid cells. Or they're going to become our lymphoid cells. Now I'll talk about both of these in more detail, but just keep in mind in the myeloid compartment, this is where you have your red blood cells. This is where you have your megakaryocyte. Which is, of course, makes platelets. And this is where you have your granulocyte, your macrophages, dendritic cells, etc. And your lymphoid cells are the three lymphocytes, the T cell, the B cell, and the natural killer cell. So let's first just discuss the development of our lymphocytes. So we have our common lymphoid progenitor cell. And this will get some sort of signal. We do know that interleukin 7 is going to drive a T cell, so you're going to generate a thymocyte. And then this is going to migrate to the thymus to become a T cell. And there is another signal, which we don't know quite, and it's going to become a natural killer cell. And we also have within the bone marrow, you're going to have a pre-B cell. And this is going to stay in the bone marrow. Oops, sorry about that. And this is going to become a B cell. So all of this you can think of is happening in the bone marrow. And then, of course, our lovely T cell is going to occur in the thymus. But our B cell stays in the bone marrow. Just to make that more clear, I'll make sure to put these guys over here. They're definitely in the bone marrow. B cells only mature in the bone marrow. Okay, so this is your lymphocyte compartment, and of course our T cells are going to be our CD4, CD8, and these are our helpers, and these are our cytotoxic. Our B cells are going to make antibodies, and we're going to have IgG, which is the dominant antibody, important for infections, many infections. We're going to have IgE, which is important for worms, for example, but also the allergic response. We have IgA, which is going to be in your mucosal secretions. We have IgM, which is good at activating complement. C prime is complement. And so all of this is very important for your immune system to keep you healthy. There is a disease that is called Xlinked SKD, and a hematopoietic stem cell transplant is necessary in order to reverse this. So SKD stands for severe combined immunodeficiency. So that's where you get the SKD. SKD are going to lack T cells or NK cells, and they typically have non-functional B cells. Keep in mind that the B cells require T cell help for most of their function, and so they are going to have a defect in being able to produce antibodies other than IgM, for example, for the most part, or they're not going to have very good even at making IgM. And so SKD is caused by what's called a mutation in a common cytokine gamma chain. And so basically what this is we talked briefly before about your IL-2 receptor. So here's your T cell. And on its surface, it's going to have an IL-2 receptor. And the IL-2 receptor is made up of 3 parts. I'm just going to draw them really big. So we have our gamma, beta, and alpha. The alpha equals that CD25, that is the target of a lot of drugs to counter transplants. The gamma here is what, is what we refer to as the common gamma chain. And this common gamma chain is going to be found on all of these different cytokines. IL-2, IL-4, IL-7, IL-10, IL-15, IL-21. And so if you have a mutation in the common gamma chain, you're not only knocking out IL-2, but you're knocking out all these other cytokines. And these cytokines are very important growth factors for T cells as well. So IL-2 is, of course, going to be a growth factor for an activated T cell. IL-7 is important for development of both B and T cells. So this is happening while the cells are developing, B cells in the bone marrow, T cells in the thymus. IL-15 is a growth factor for memory cells. IL-4 is a Th2 developing cytokine. IL-10 is important for T regulatory. And IL-21 is important for Th21. So those are the bacteria in your gut, for example, things like that. And so all of these processes are going to be impacted because you don't have the gamma chain. If you don't have the gamma chain, these receptors that either bind to IL-2, IL-4, IL-7, etc., are going to not be able to signal. Now keep in mind the gamma chain is the link to all these cytokines. These cytokines all have different alpha chains. Some might have a beta chain too. Some do, some don't. But they all share the gamma chain, but not the other components of the receptor. So it makes the receptor specific for a specific cytokine. But if you have a mutation in the common gamma chain, because all these cytokines have to share that common gamma chain, it impacts a lot of your immune system. Now, in order to do this, you can use stem cell transplant. And so stem cell transplant will try to transplant what we refer to as CD34 positive stem cells. These are the pluripotent stem cells. So those are those stem cells that are self-renewing and can become any of the different blood cell types. And you can transfer the stem cells. There is a process of acceptance versus rejection, and we'll discuss that more when we talk about transplant immunology. Okay, so now let's go to our myeloid. So those are our lymphoids. So now we have our myeloid development. We have, just like we had with our lymphoids, we have a common myeloid progenitor cell. And the common myeloid progenitor cell can become many things. Now, if you're going to make red blood cells, the trigger for red blood cells comes from the liver, and it is erythropoietin. Then you'll get your erythroid progenitor. Which will become your red blood cell, or your erythrocytes. You can also have thrombopoietin. And thrombopoietin is also referred to as platelet growth factor. And this will give you your megakaryocyte. which will release platelets. And then you have your myeloid. You have your myelomonocytic progenitor cell. And we'll continue with this one on another page. This kind of looks like a macrophage. And this is going to be caused by some growth factors. And this is GM-CSF. We have G-CSF and M-CSF. So GM-CSF is granulocyte monocyte. Oops. Colony stimulating factor. And then GCSF is granulite colony stimulating factor. And then MCSF is monocyte colony stimulating factor. And so that's just what those terms mean. Now, so when you have GMCSF, GCSF, and MCSF, they're going to drive that myeloid progenitor to become a myeloid monocyte progenitor. And then this will differentiate into additional cell types, which we'll talk about. Now, if you only have GM-CSF and some cytokines, so we have GM-CSF, so your granulocyte monocyte colony stimulating factor, plus IL-3, you're going to drive towards your eosinophils. So this is going to give you a progenitor with IL-5, and you're going to get an eosinophil. Eosinophils are like neutrophils. They have lobed nucleus. And so this is a granulocyte. And they're going to have granules. And if you have GM-CSF and IL-3, oops, sorry. I just keep doing that. Sorry about that. By something else, not sure what it is, you get a mast cell. And these have big granules. Now keep in mind, an eosinophil is going to have a lobed nucleus. A mast cell will have larger granules, not a lobed nucleus. They both can be from the myeloid progenitor cell in the presence of granulocyte monocyte colony stimulating factor with IL-3. But then they have to further differentiate. Eosinophils are going to further differentiate with IL-5, interleukin 5, and the further differentiation of the mast cell is not known yet. Okay, so then we also have that myeloid monocytic progenitor cell. So let's draw that guy. And now this cell is going to give rise to our neutrophils. So this cell is going to, with, again, GM-CSF plus IL-3, you're going to get a precursor, which through granulocyte colony stimulating factor only, you get a neutrophil. It's going to have a lobed nucleus. And then if you have GM-CSF, with your IL-3 and monocyte colony stimulating factor. Let me actually do it on this side. Then this is going to lead to the branch that makes your macrophages and your dendritic cells. So you can get a monocyte, and monocytes travel in the blood, and then when it enters tissues, it becomes a macrophage. And then you also have another branch, nucleus, and this is going to become a dendritic cell. And this is through GM, CSF, still present, and TNF alpha. TNF alpha is tumor necrosis factor, alpha. And so it's identified, of course, in tumors, but it is a growth factor for, in this case with hematopoiesis, our dendritic cells. All right, so the last thing I want to discuss on this topic is what's referred to as a shift to the left. And the reason I want to talk about this is because it's a common term used in clinics, and it refers to the presence of immature blood cells in your circulating. So you have immature versions of your blood cells in your circulation. So keep in mind that all of these blood cells that we just talked about, your neutrophil, your dendritic cell, monocytes, etc., mature in the bone marrow, and then they are released as mature into the bloodstream. And so a shift to the left it's called a shift to the left because you always start left, you go left to right, and over here is the most immature, and over here is the most mature. And so there'll be some sort of threshold where you'll have bone marrow, and then you'll have circulation. Now, in your normal circulation, you will always have, with respect to your neutrophils, multi-lobed neutrophils. So over here, you're going to have several different stages. But in essence, they all look kind of multi-lobed. So you can have and then inside of your bone marrow is where you typically see your more immature. Oops. Let me. Don't worry about the size, that doesn't do anything. So your most immature has this giant nucleus. Doesn't even look like a neutrophil, just has a very large nucleus, and that's a myeloid blast. So it's blasting. Blasting just simply means it's replicating. And then you go on to your promyelocyte, to a myelocyte. Let me write it up here. to a metamyelocyte to a band cell. And so here's our band cell. And our band cell is just what it is. It looks like it has a single nucleus, and it looks like a little band. Now these stages here, the nucleus is slowly starting to form into a band cell. And then when it gets released, it becomes a mature neutrophil. Which is segmented. And segmented is referring to the nucleus. And so when you talk about a shift to the left, we have over here a gradient you can think of as a shift to the left. So we have our most severe, and then we have sort of a moderate shift to the left. And then we will have just what's referred to as normal. So if you take someone's blood and you see a whole bunch of band cells, that would be a moderate, mild to moderate shift to the left. Now, if you take someone's blood, you don't see any band cells and you don't see any segmented neutrophils. All you see are very large myeloblasts and promyelocytes, metamyelocytes, then that would be a severe shift to the left. Okay, so that's all. Thank you.